Hi everyone, my name is Mohammad Lotfollahi, and today I'm going to talk about learning large-scale perturbation effects in single-cell genomics. So by the availability of large single-cell atlases, specifically perturbation atlases, we can use these reference atlases to predict a couple of downstream tasks. So the main task is centered around predicting the effect of a perturbation, but it breaks down into multiple tasks, like what to actually predict. You can predict predict an omic, so basically the effect of a gene expression, for example, after a certain perturbation. You can predict phenotypic measurements, such as area under curve, or you can classify targets and mechanisms, or you can classify and predict interaction prediction. So basically, how two different drugs interact with each other, or how two different genetic knockouts, and similarly for chemical property prediction. So among these tasks, today I'm going to talk about omic measurement prediction, specifically gene expression prediction, and the motivation for that is so single cell experiments with all these advances is still an expensive technology. And if you want to use single cell for drug discovery, specifically for combinatorial drug discovery, it's hard due to that the nature of drug combinations. So basically you have combinatorial explosion, and also these single cell technologies being expensive. So to address all these problems and provide in silico prediction and guide experiments, we came up with new model called Compositional perturbation autoencoder. It's a general model, but we try to predict single cell effects using this model. So the model receives gene expression as input. It tries to disentangle the effect of drugs, covariates. So covariates basically can be cell lines, cell types, patients, all the discrete covariates, to come up with the basal state, which basically has none of these information. And this is done using discriminator, like having different discriminators and adversarial training. And having this basal state, we learn to linearly add back the effect of perturbations and covariates. So for each perturbation, we learn a drug embedding or perturbation embedding, similarly for covariate embedding. And then we have nonlinear dose, and then we add all these effects as a linear effect in the latent space. And then we can predict the effect of a certain perturbation with certain covariates and dose and time. So basically with this model, you can predict novel conditions. You can provide dose response modeling due to explicitly modeling dose. And then because these embeddings are learnable and then they encode the similarity of different perturbations, you can visualize these different embeddings, for example, for drug repurposing studies, because they provide similarity between different drugs. So I'll show you an example of the first application where we actually tried our model on a large-scale high-throughput single-cell experiment from Cold Trapner Lab, where we had 3 different cell lines, 188 drugs in 4 different dosages, with like close to 500,000 cells. As you can see, this is the UMAP of the original data, and apart from this, colored by the different mechanism of drugs. And as you can see, most of the drugs are basically mixed except one epigenetic drug. So overall, this is like a super mix, and you cannot figure out something from looking at the UMAP. So as an example, we held out one drug at the highest dose, and then we tried to predict that gene expression, all the genes for that drug. And here you can see as an example the predicted genes for that specific drug in the highest dose, and the model actually picked out that it has to upregulate those genes. Similarly, it can pick out the model can actually pick up cell type specific effects, where you can see it figured out that these 2 drugs should be effective on this cell line, and the blue drug here should not be effective. So basically, it picked out non-responder cells. And finally, we provided like more comprehensive benchmark where we showed that as you leave out more drugs and more cell lines, the predictive power obviously gets worse, but overall it has a robust performance. Here is the cell line latent space and also drug latent space, where you can see that similar drugs with a similar mechanism of action. They're clustered together, and this drug latent space can be used for downstream tasks such as drug repurposing. Here, the most effective drugs are epigenetic regulation, and you can see our nonlinear dose encoder also picked up this effect, and the dose for these drugs are actually have a bigger size effect. So the second example, we applied our model on a single cell knockout experiment with genetic knockouts with 287 single and pairs of knockouts. And here is our genetic manifold, so knockout latent space. And you can see that the model grouped similar knockout inducing similar gene programs into close by clusters. So similar as before, we held out a double knockout, and this double knockout has synergistic effects, and we showed that the model actually can predict these synergies. Similarly, it can predict epistatic interaction, where actually one factor completely masked the other factor, and the model actually picked up that effect, while it hasn't seen the specific combination in the training data. And finally, and the most importantly, we imputed and predicted all the missing combinations for this specific data set. So here you can see drug latent space or perturbation latent space from measured and the predicted ones. So we imputed 5,613 missing double knockouts, and we generated single cells for them by providing an uncertainty. So as you can see, as we go from points that are far from the training points, our uncertainty gets higher. And then we annotated all these missing and predicted combinations, and we found out for most of them there is one factor that dominates all the points in that specific cluster. And we zoomed into one of these clusters where we predicted all the interactions with KLF1 with the second gene, and only small portion of them were actually measured in the real data. And going into the cluster, we found out all these predicted double knockouts, they are from different interaction regimes, such as synergistic, dominative, and all these different things. So the model actually picked up the effect, different effects. So overall, I showed our model toward perturbation atlas for the using perturbation atlases, and we assume that there will be more work on incorporating molecular priors and spatial information. And finally, I would like to thank my collaborators, and I would be happy to take questions.